I'm Roger Alexander, the Scientific Outreach Coordinator for the Extracellular RNA Communication Consortium. Welcome to this month's ERCC webinar. Today we have Dimitris Lagos from University of York, from the Hull York Medical School there and the York Biomedical Research Institute. He's an immunologist who studies the role of non-coding RNA. And today he's going to tell us about maybe some of that background, but also specifically about the role of non-coding RNAs in COVID-19 and his research with the UK Coronavirus Immunology Consortium. If you want to put questions in the chat, we'll ask those first at the end of the talk, and then we'll open up the lines. So Dimitris, we're looking forward to hearing about your work.
Okay, yeah, thanks, Roger. Nice to see you all, and thanks for the invitation. What I'll tell you is a little bit about our work with coronavirus. So I thought I'll start by telling you where we are. We are in a small city in the north of England called York, and the houses look, not all houses look like that, but some look like this. We have a big church that we call it a minster, not a cathedral. And this is a very small city generally. And we are based here. This is an immunology center. And we cover a lot of different contexts in terms of infectious diseases, in terms of the link between hematology and immunology. And in my lab, the main interest is how non-coding and regulatory RNAs affect immune gene expression. So I'm not going to talk about this today, but just as a taster of what we do, where, for example, we work a lot with long non-coding RNAs and their interacting proteins. And Katie is one of the people who's leading this project in collaborations with the Crick Institute and the Wellcome Sanger Institute. For example, how MALAT1, a very well-known long non-coding RNA, how this works in immune cells. Another really interesting project we have as a taster, again, is using long-read sequencing to understand and, to a certain extent, redefine cancer transcriptional and post-transcriptional profiles and how these affect interaction with immune cells. But as I said, these are the more mechanistic things we do in the lab, and other examples of similar type of work around non-coding RNAs, their interacting proteins, and how these affect outcomes in terms of immunity, either to infection or to cancer. So today I'll tell you about this type of work that we're doing, which is essentially trying to study non-coding RNAs or measure non-coding RNAs in people and try to use these measurements to understand disease. So the example that obviously is it was a very topical and is still a very topical example is around SARS-CoV-2 and COVID-19. So this for us started by being part of a very large consortium in the UK, which was essentially put together as in sort of an emergency fund, where brought essentially all immunology centers together. It was a very short, as I said, emergency sort of type of work, where the remit was, let's get all the UK immunology together to try to work out some questions around COVID-19. It was chaired by Paul Moss and Peter Openshaw. And as I said, there were 20 participating centers, including research institutes, universities like ours. And we had a lot of support by our Society for Immunology in the UK, which also led a very interesting experience, which was talking to public and patients and engaging with them about our work as it was actually happening. So the aims of the UKCIC, as we call it, the UK Coronavirus Immunology Consortium, was essentially to understand, first of all, what are the determinants of susceptibility to primary infection, how protective immunity, either B cell or T cell immunity or innate immunity is generated. There was questions around pathology. What is the deciding factor that determines, or factors that determine pathology? And then some more virological and host pathogen questions around cross-reactivity, for example, of coronaviruses, so SARS-CoV-2 with a pre-existing coronaviruses, or more, and an interesting aspect actually, are there any mechanisms, what are these mechanisms of that the virus escapes the immune system? So these questions were turned into themes within the consortium. So we have 5 themes, and different groups were assigned to different themes. So this included, as I said, the primary immunity. So how do we understand the primary response to COVID-19, and how this relates to the outcomes? Protective immunity, immunopathology, cross-reactivity, and immunization. And actually, the work we did in York, here in the UK, in our university, was mainly around theme 1 and theme 3. The work I will present today is mainly our contribution to the primary immunity theme of the consortium. So we started this, and actually we got into this, and there was even at the very early stages of the pandemic, when people were talking about some sort of cytokine storm, or some sort of elevated cytokine levels in people who go to the hospital and are very unwell, or they need a lot of intubations. So this was a very early concept. This is a type of cytokine storm, perhaps not identical to other lung viral infections, but similar. So this is demonstrated here. We would take one of the very well-known cytokines, IL-6, and you see as the severity of the disease increases, levels of IL-6 increase. But one thing that was very interesting, and so this is from one paper, but there are, as you probably all know, there are several papers, large-scale studies that did similar things. No matter which one you look at, there is this observation, there's a gray zone here. So these are people who have essentially in their circulation the same level of cytokines, yet they present with very, very different severity of disease. So this is the World Health Organization scale of the disease. So 3 to 8, we're talking about someone who just needed some oxygen versus someone who actually is critically ill. So this gray zone was something that from the very early stages, and as someone who's been working on non-coding RNAs and more on microRNAs a few years ago, that caught my attention that maybe there would be better ways to, or different correlates, systemic correlates of infection and disease that could help us sort of separate these groups a little bit better. So the question in simple terms is, why do patients with similar cytokine levels present with different disease severity? Or how can at least find things that are actually different between people who have different severity of disease? Before I go forward, I have to say that this work obviously involved many people. I'll come back to that. But the 2 co-leads of the work were Natalie Signoret, who's a cytokine and chemokine expert, and Julie Wilson, who's a professor in maths here. And she was the person who kept us in tech, basically, on how to analyze the multiple data sets that we acquired. And the rationale essentially was to measure circulating cytokine and chemokine levels and circulating microRNAs in the same patients, and trying to simplify, and yes, this might be a little bit of an oversimplification, that if we consider cytokines and chemokines as perhaps instigators of pathogenic signaling through their interactions with the receptors, then we could consider microRNAs as indicators or at least regulators of intracellular gene networks, which might operate in cells that are mediating the response or responding to these inflammatory stimulants. And the useful bit about that is that, of course, that both cytokines and chemokines and microRNAs are detectable in plasma or blood plasma or serum, and we use blood plasma for our studies. So one thing that's probably very, very relevant to your consortium is that we didn't purify exosomes. We didn't do anything fancy like that. We just took blood plasma and measured cell-free microRNAs, essentially. So the first part of the study was in collaboration with Manchester, which is a big city here, has many hospitals. So this was collecting samples from 3 hospitals from the very first wave of the pandemic, where essentially these were samples that were collected for research. And our collaborators there had already had a collection of plasma samples that they sent us to do cytokine and chemokine multiplex profiling at the same time of doing microRNA profiling. So we did microRNA profiling in 171 plasma samples. These were the profiling, the platform we used was the nanostring platform. And then the idea was to measure for every single sample both cytokine levels and microRNA levels and see if we can get anything informative there. Sorry. The way we split our participants, the patients, were based on the way that they were treated. So we didn't use, for this particular study, we didn't use the World Health Organization classification. Rather, we split them in these groupings of mild, moderate, and severe disease. And that was based on the work that had preceded our collaboration with the groups in Manchester, which is published in Science Immunology. It was one of the very first papers on COVID-19. So the first thing we wanted to do was to actually see, can we identify severity associated microRNAs? So can we identify microRNAs that are associated with severe disease? And so we did that, and as you can see, some the red ones here, the ones that reach significance after correction. And yes, there are some microRNAs that can be upregulated or downregulated in people who have severe disease. And actually, if you do essentially the equivalent of gene ontology, but for microRNAs themselves, we did see some interesting things. For example, an association of microRNAs that could be associated with cell death, or interesting things like vascular inflammation. So terms, functional terms that could be, they were relevant to a severe disease like COVID-19. The thing that was interesting here was that even though we're detecting this in the periphery, in blood plasma, there were not many sort of immunologically relevant terms that were coming up, apart from chemotaxis and innate immunity, which made us think that there might be something around the fact that these microRNAs we detect in circulation might actually reflect the tissue state of people with the disease. We did something similar when we reverse the question and look at the mild patients, people who had milder disease and still hospitalized. And I'm showing this because there was something really interesting about this. And that was that essentially we saw significance going one way here. So people with mild disease, we found microRNAs that were upregulated. But actually, if you look at these, you'll appreciate that this is very skewed, which made essentially what this suggests is that as people progress to moderate or severe disease, perhaps there might be lower levels of circulating microRNAs in the periphery. And actually here, I'm showing a different type of analysis rather than taking the microRNAs themselves. We took their predicted targets and again looked at a sort of functional associations and enrichment. There are a lot of terms here to do with RNA processing. So these are the targets of these red microRNAs, as you can see. This might be interesting in terms of what it's telling us. Obviously, the virus is an RNA virus itself. It might be also an indicator, it might correlate somehow with the fact that to this decrease that we see in circulating levels of these specific microRNAs in moderate and severe samples. I'm just going to show one thing about the cytokines, just to show that to give you an idea of the complexity here. So we, at the same samples, exactly the same samples, we were looking at these cytokines and chemokines, and we do see some clusters that other people have identified as well of correlations operating in all groups, so that they are valid in all groups. It's the thing again that I was telling you when I started about this idea about the cytokine storm is reasonable, and it's definitely happening, but in the periphery, you might not be able to pick that up in terms of being able to tell you who are the individuals who are more likely to develop severe disease. But even, and this is essentially the preamble to the couple of things that I'll tell you about microRNAs, but even with cytokines, we started seeing what we call as qualitative differences, not quantitative differences, not more of the same happening in as severity progresses, but correlations, for example, that only exist in one of the groups. And this is a very striking one. We take one inflammatory cytokine, interferon gamma, and CCL20, a chemokine that plays a very important role in respiratory infections. What we saw is that in the severe group, these two correlate positively. We don't see that in any of the other groups. These are COVID-19 hospitalized patients from the first wave. So a group of people who needed to go to the hospital, yet these correlations seem to be happening only in the severe group, which obviously it's interesting if you're into these type of things, but it also gives us the idea that there are some qualitative differences, so some step changes between severity groups. So one other thing that we want to do when we talk about the patient classification or stratification, we wanted to see if we combine cytokine and chemokine profiling, which we call CC, with microRNA profiling, whether we can actually start separating people with severe, or samples from people with severe disease a little bit more from the rest. And actually what we were finding was that if you combine both modalities, you do get better separation, actually, in terms of how accurately you can predict if a sample is comes from a patient with severe disease or from the rest of the group. And it was actually better than using either microRNAs or cytokines and chemokines alone. Again, telling us that we were going, we were moving towards the right direction. And then it comes to the thing, I started and starts with IL-6. Then we asked the question, if you take microRNAs and say, what are the principal microRNA correlates of IL-6 levels in the periphery? So we say a patient comes up and has high IL-6 levels. What is the microRNA baggage that goes with it? What's the microRNA correlates that go with it? And one of the things that is interesting in this is that you might be able to read these, but these are different microRNAs that would be the primary correlates of IL-6 in different severity groups, suggesting that when you combine microRNAs with cytokines, you start seeing these very clear qualitative step changes between groups. So, for example, a microRNA that we've all have come across at some point in our lives, if you work on microRNAs, microRNA 30 here, one of the family is negatively correlates with IL-6, but only in the severe group. And actually, when we looked at some other cytokines that are very, very important in driving the pathology of the disease, we got some things like that, which were, in my opinion, they were very, very striking, because it's not only that you see different microRNAs being the principal correlates, say for CXCL10 levels in the periphery, it's even different it's a different direction of correlation. So here we have 5 principal correlates, and they are correlating positively with CXCL10 only in severe patients. And this gave us, and so similarly for, we in the paper actually we have that for every single cytokine and chemokine we tested. And this, of course, is correlative, but what it shows is that there are some very clear separations between the groups. So, and perhaps saying that IL-6, having IL-6, high IL-6 levels is bad, might not be as informative as saying having high IL-6 and low MER30 at the same time. This is actually a stronger indicator of severe disease. In the paper, we also looked at some of the principal blood measurements that people will have done in the hospital anyway. And for example, the lymphocyte to neutrophil ratio is a very well-known correlate of severe disease, or CRP, a C-reactive protein, which is, again, something that's measured in all patients. And we identified the principal microRNA correlates for all these factors. And actually, again, in some cases, we see these very striking differences in not only which microRNA correlates with these parameters, but also the direction of the correlation, such as the lymphocyte correlation here. So lymphopenia and neutrophilia is what characterizes people with COVID-19. And one last thing that we did from this first cohort was an interesting sort of analysis, which is called canonical correlation analysis, where we did, when I showed you the microRNAs that were actually the principal functional annotations, we talked about, for example, that there was a group of microRNAs that were enriched in cell death functional annotation. By finding which cytokines are correlated with a specific group of microRNAs, we started getting an insight of which, say, cytokines, for example, here, could be the principal correlates with cell death in COVID-19. This is essentially taking microRNAs and cytokines and trying to identify what are the right pairings, but in the context of a functional annotation. That would be very hard to do for cytokines, because if you put your cytokines in any sort of gene set enrichment or annotation, it will come up as inflammation, and you won't know any specific pathway. But having the microRNA information gave us a handle to start connecting some group of cytokines to actually more specific functional annotation. So for this first part, essentially what I think we were trying we contributed a very, very small, obviously, contribution compared to the amount of work, was towards this idea that COVID-19 seems to be, and other work has shown that as well, in using different modalities, different measurements, it's a stepwise disease. So it's not a kind of continuous progression disease that everything happens more of the same and you can catch it at every level. There are some specific step changes that are associated with the switch from mild to moderate and moderate to severe. And actually, these might be the areas that we should be concentrating in terms of therapeutics, current or future, for COVID-19, essentially what pushes you over to the next step. So in this first part, I mentioned some ideas about how we integrated plasma, microRNA, and cytokine and chemokine profiles to identify severity-specific hallmarks of the disease. We found specific combinations of microRNAs and cytokines and chemokines that would underpin clinical correlates of the disease, and a microRNA signature that was distinguishing severe cases from the rest. Actually, there was a reasonable but not complete overlap with studies that had been done before us, where they were just comparing infected versus non-infected people. Not this question about severity, but it was controlled versus infected people. The second part of the story was where we wanted to ask this question. Okay, we do find microRNAs and cytokines that are differentially expressed in samples that were collected for research, frozen very quickly, people working day and night to be collecting these and getting them to the right state very quickly. Could any of these be useful in terms of a clinical or a hospital or a pipeline? So then we did something slightly different. So the normal route of what happens in the UK, you're admitted to an A&E, so this is the emergency department in the US. People present there, and there would be a sample, a blood sample that's being taken, and this is going to be used for your complete blood counts, so things like all the diagnostic and labs that you do in the hospital, and by law in the UK, this sample has to be kept for a certain amount of time, a minimum of 6 hours, in case something has gone wrong with this blood count, so it can be done again. So what we did was to say, could we just take that sample after it has been used for normal routine care of the person who came to the hospital, isolate the plasma then, and try to see whether we can detect microRNAs or cytokines and chemokines. So this happened in our hospital here in York, which is a much smaller hospital than the ones in Manchester. It was what we called in the UK the second wave. It was the second wave everywhere with different timings. And what we call, these samples are generally called leftover samples. So because they are kept essentially on the bench in case they need to repeat that first CBC for people who have come to the hospital. And the good thing about that was we caught everyone their first sample as soon as they essentially, if we can say that, cross the door or come into the hospital. So this was another large number of samples, 166, and we did again the multiplex cytokine and chemokine determination. And here we looked at some selected microRNAs that were selected from our first study in the Manchester cohort. Again, this story about cytokines being important, but some of the signatures are maintained through severity. And actually, if I'm honest, one of the big surprises here was that even though these samples were what we call leftover samples, we could detect cytokines and chemokines, and actually we could detect a lot of the same or very, very similar patterns to what we saw in the first study with the samples from our first cohort. And in terms of microRNAs, even in the second independent validation, and actually an interesting thing there about the first and the second cohort is that we were looking at even slightly or drastically different variants of the virus itself. We did identify microRNAs that again were more highly expressed in samples from people with severe disease. So here I'm giving two examples, microRNA 4454, and this was the top severe disease-associated microRNA in the first cohort. It's interesting that some of the targets of these microRNAs are immunologically relevant genes, or some of the predicted targets, should I say. And microRNA 630, which kept coming up in our study quite a lot. And again, some of its targets do include inflammatory and either cytokine receptors or cytokines themselves. This is just an observation, but it is interesting thinking ahead whether these are maybe microRNAs we should be looking at more in terms of mechanistic studies. And one thing that this study allowed us to do, because it had even though it was a similar number of samples, there were more patients in this study because we had fewer follow-up samples. The one thing that this allowed us to do was to start asking the question, can we identify parameters that are associated with outcome? An outcome here was either, unfortunately, death or people being discharged from the hospital and going home. And that was actually very interesting here in terms of that we did find some things that people might have expected. For example, in the top left graph, IL-6 was coming up as something very significant, and IL-10 associated with bad outcomes, unfavorable outcomes. In terms of chemokines on the bottom left, CCL20 was very clear. If you see this, its distribution. And in terms of microRNAs, we did see that some of these microRNAs that we were measuring were actually associated with poorer outcomes. And essentially, the message here that elevated levels of some of these cytokines and decreased levels of microRNA 495 or 451 or 29B were associated with a fatal disease, These are different ways of presenting that in terms of principal correlates of outcomes. These essentially tell you if you have, we split the samples as high levels, top quartile levels, and we see that there is an overrepresentation. For example, if you look at the microRNA part here, high levels of these microRNAs, you would find them in people who are more likely to be discharged. And then you do the reverse where you talk the bottom quartiles and you see that low levels there, the cytokines perhaps are more important. But what this allowed us to do is essentially start saying, okay, can we start building some models, some forward regression models where we start asking what are our best parameters that could be associated with the outcome. And we started by CCL20 alone because this was one of the most significant when I showed you all the graphs, all the data, and then start adding other factors. And so as it's not a surprise, but I think when you get into this 80–90%, this is perhaps very, very good in terms of being able to separate people by outcome. These last two rows here, residual deviance and AIC, these are essentially, they're telling you how good your model is and how much overfitting and things like this you do. And the lower this number, the better in terms of how good your model this is. Again, you see that as we are increasing the accuracy, these parameters don't get worse. If anything, they get much better. This, whether we're comparing how good CCL20 would be compared to adding some principal cytokines and microRNA 451, or having a signature of 8 cytokines and 3 microRNAs, it was giving us some idea about how we can associate these peripheral parameters that we measured in blood samples that were really taken for normal routine care. We could start getting an idea of people who are more likely not to have a very good outcome. So in terms of these parts of the work, the thing that I must say, we didn't know, I was quite confident about microRNAs, if I'm honest. I wasn't quite clear about how cytokines and chemokines would survive a sample being left for a while and being kept for because it's a legal requirement to be kept there. But we could detect them, we could detect very similar patterns to what we had in our first independent cohort where the samples were collected for research. And I suppose a good outcome from this work is that we started identifying factors that are associated with fatal disease. And I think that is useful, especially as we're going forward, and we have gone forward, where even during peaks of the infection, we will be seeing a lot of people who will be going to the hospital, but hopefully and fortunately, not as many of them would need a very strong treatment. So the earlier you can have some tools to be able to triage patients or help with decision making in the clinic, the better, I think. And these type of things might be more useful now as we're getting, hopefully we will get to a point where fewer people will need a lot of attention to be able to identify them as early as possible. Some of the things that we currently do, and they are not published yet. So we are trying to look, okay, we try to take some of the work we did and published and say, okay, can we combine some of these indicators, correlates of severity with things we measure anyway in the hospital, and develop sensitive, specific, and affordable bedside tests that we could integrate, or at least in principle integrate with our health system. We are doing, actually we just finished this bit where we're looking at some of these microRNAs that we identified as severity associated. We used an approach where sampling from the nasal cavity is called nasosorption. We got some samples there from yet another cohort from London. And actually, we could detect some of these microRNAs even in the upper respiratory tract of people with disease. This is more starting to address where these microRNAs come from, and we find them in circulation. And another part which ran completely in parallel, and we haven't, we are close to writing this up actually, is looking at our other favorite non-coding RNAs, long non-coding RNAs, and how can this help us understand the pathology of the disease. So we had a collection of post-mortem lung sections that we analyzed a lot of single-cell sequencing data there, and I found something really interesting, associations or little bits of information about specific non-coding RNAs and how these can actually help us understand the type of pathology and the type of immune cells that make it to the lungs of people with fatal disease in this particular case. So that's me. There are many people who, as you can imagine, there are many people who have helped with that from hospitals, from collaborators here, people who worked under very difficult circumstances. Our technology facility here, people who were running these hundreds of microRNA profiles, clinical collaborators in Munster, Newcastle, Imperial, our maths person here, Julie, who was amazing analyzing this data. And our funders, the Medical Research Council and the Wellcome Trust. And I always have to be, I am very grateful to people who agreed to give samples for these studies or their families. These studies would never, in any of the ones that we've read, would never happen if people were not so keen to help medical research. And thank you for your time.
Thanks very much, Dimitris. There's a couple questions in chat. Meena Kumari asks, how many microRNAs did you identify in the plasma of COVID patients?
So how many we were confident in terms of identifying? I think the number was around 100 or 80, something like that. We, as you probably know, it depends on how harsh your threshold is. So we had essentially we set as a threshold microRNAs that were detected in. So if we had microRNAs that were detected in most of the patients, not only about the level of it, but being detectable in patients. So the second question I can see here, which is about whether we have identified, looked and compared to infected cells. No, we haven't done that yet, and we are approaching this from a slightly different angle, which is to, and that's why this upper respiratory tract study is very interesting, is to actually see if we can detect them in epithelial cells of people, and our access to some of the post-mortem samples, albeit these will be only very severe disease, could tell us even whether we can detect them in lungs, and then this, in lungs of people with the disease, and even with cell types. I must say a lot of the microRNAs we found are severity associated, as much as you can say these types of things. To me, they were microRNAs that were known to have function in epithelial cells rather than immune cells. That's what makes me think that essentially these signatures we're picking up in the periphery, they are mainly telling us what's happening in the affected tissue rather than being telling us what the immune cells are doing in the periphery. And that's, of course, in oncology, that's done very, very well, because people have done very elegant studies comparing tumor microRNAs with circulating microRNAs and see that this is the case. Yeah, I think we are looking into the source of these microRNAs. I don't think that these are viral microRNAs, because I'm pretty confident, at least the ones we measured both by nanostring and PCR, that these are human. Actually, the one that we found a lot of difference for this 4454, I think, if I remember well, this is actually human specific, or primate specific. So this again tells you something about how different species can react to the infection.
Sort of a follow-up on the question about, have you taken things into cell lines to look for mechanism? You showed there's like a step change. There's mild, moderate, severe. It's not a continuous worsening of symptoms. Do you have any sense of, what the biological, what tissues are getting hit? Is there some specific biology that's turned on in severity? Is it like once it gets into the lungs, something like that?
Yeah, I think there's definitely a combination of reasons, and there's a lot of elegant work on this. And I think how obviously this is a lung pathology, and how the immune response to it is one part, how this is regulated or resolved. But I think there are all sorts of experiments I'm trying to remember. So people have shown that with proteomics, blood proteomics. They have shown that with obviously a lot of single cell sequencing, that especially in the earlier stages of the pandemic, essentially the whole thing, by the time people were going to the hospital, it was already shown, the dice was cast, as someone said in the talks that I went, as to whether the disease would be severe, moderate, or mild. So there are all sorts of interesting things that have come up since then in terms of what are the determinants of this. But yes, certainly lung pathology is the driver there, I think. So there's, yeah, sorry, Roger, sorry.
Oh, I was going to say that Minu has a question about MALAT1, but I want to continue on the COVID front for a bit and then ask that later. So I'm curious about these leftover samples. First of all, in your work, I presume you don't have any healthy controls. It's like severe, moderate, mild, on the comparisons between those. And I'm wondering what fraction of people who go into the hospital and get that blood sample taken, what fraction of them give permission to do research? And it seems like a really more generally useful resource for people developing liquid biopsies for all kind of diseases. Is there something formal in the UK about making that available and trying to turn it into clinical diagnostics?
Yeah, of course. I mean, I think that different health systems have different sort of rules, but I think they all have some rules. And in the UK, some of the samples were through the consortium were available to partners in the consortium. The study we did here is so for every study you have to do the ethics, and then depending on how you do your ethics, you have the sort of ability to share or not share. There were some things that if something is basically sort of set up as a biobank is very different as if it's set up as a specific study. And in the UK there is an amazing resource called ERCC for saying run from Imperial College that was going on for many years about respiratory infections way before COVID, about flu, RSV, and they were obviously one of the first group to get into this, and because they had a lot of the infrastructure, thankfully. Yeah, I think this is the thing you say about not having healthy controls. I think this was partly, it was a lot by design, because we wanted one thing in my mind that was very difficult, especially in the first, when we started doing the study, was to separate exposure from someone not having symptoms but having been exposed to the virus. Because remember, the first wave, we didn't have tests, we didn't have any of these things to know who is positive or negative. So there are other studies, and as I said, there is some overlap between studies that looked at this healthy, or what you want to call it, non-infected versus infected, and microRNAs we found associated with severe disease, suggesting that some of them are just a direct outcome of infection. Yeah, I think our question was try to look at hospitalized patients only and try to see, are there any differences there? There is this question about, actually, I want to go back to your colleague Pinar, about the viral microRNAs. When my training was on Kaposi sarcoma herpes virus, one of, I think, probably the first one to show microRNAs, viral microRNAs, and actually that's when I started working on non-coding RNAs and microRNAs, and somehow always try to be a little bit away from the viral part. Of course, you probably are very familiar with discussions about as to whether RNA viruses can have a lot of microRNAs for several reasons there, about how the RNA is used. Must say, we can have a look, because I know there are papers now, there is work in predicting or validating some potential SARS-CoV-2 microRNA, see if there is similarity between some of our top candidates. That's a very good idea.
This may be a stupid question, but is there any, I mean, this sort of standard PCR test, is there any correlation between viral count and severity? And related question, do you have any sense yet of how prospective your cytokine microRNA profiles are? You know, is it when somebody has this severe situation, you see it, or is it like 6 hours ahead, or, you know?
So one thing that was interesting was that for a lot of these patients, especially in the first cohort, we had follow-up samples, and in our study we couldn't see any temporal patterns of these microRNAs, as essentially for the time that people were in the hospital. There may be many reasons for that. So one of the things that we ended up doing actually for some of our analysis, we were either taking single time point, the point of presentation, or an average from all the samples we had available per patient, as a more stable sort of indication of microRNA levels over the period they were in the hospital. And actually there was a lot of similarity there in patterns, some differences. Yeah, I think it's hard to say. I don't think there is a very strong correlation with viral load in terms of severity of disease. Obviously, there is some, but you still always have these gray zones that attract me these and display minds. Do these patients? Yeah, that's a great question, but we haven't looked at the vaccinated people. These were the first and second wave, so this was before vaccination came into play in the UK. But yeah, that's a very interesting question. How do we essentially look at vaccinated people? I must say, at least in our hospitals, even in the most recent wave of the disease, most of the people who were in the hospital had not had the vaccine, so some of these would be still relevant. But yeah, going forward, these questions, I think a positive outcome from that was to actually to validate the concept that you could find correlates of disease that were qualitative correlates, and I think that could help. Yeah.
Did you want to take up Mina's question about MALAT1?
So yeah, MALAT is it's one of our favorite children currently, and we work extensively on it. So we have already one paper out on MALAT and its role in T cells, in T helper cells. It's I can talk for a long time, but it's an amazing sort of situation. It's one of the most, as you probably know, most of highly expressed transcripts in our cells, perhaps outside the ribosomal RNAs. It's always within the top 5 if you do your RNA-seq, if top 5%, sorry, not top 5, top 5%. And it breaks a lot of the rules of non-coding RNA, so it's conserved, relatively conserved in mammals. It's very highly expressed, 5,000 copies per cell in some contexts. And then you can knock it out, and mice, at least mice, are absolutely fine. And again, having trained through microRNAs, where people thought the idea that a lot of this post-transcriptional gene regulation is relevant in stress, and being an immunologist, we thought that stress is infection. And actually, that's how we got to work on MALAT in T cells. It's very quickly suppressed upon activation of T lymphocytes. And we actually showed in 2 different models that yes, it is suppressed during activation of T lymphocytes, but if you take it out completely, you have very strong responses. And sometimes this is a good thing because it can clear the pathogen. Sometimes it's not a good thing because it can promote immunopathology. So we use the model of malaria, where an unhinged immune response can cause severe pathology, and we saw that effect actually. Since then we've done a lot of work about its binding partners, adapted some methods in identifying binding partners in primary T cells. Also what its effects are on its binding partner, so in turn, so it binds a lot of RBPs, as you know, and then what it does to them. Yes, and then we are continuing on this, and yeah, I'm happy to discuss outside here if there are any ideas about it. It's an amazing entity within every cell, and yeah, it's I think there's still things we can learn about it.
We're near the hour, but I gotta ask one more question. Or actually, I have a suggestion. So you showed those heat maps of microRNA expression in the three cases, and I think it would be interesting. I know that there's sort of trees underneath them, but it would be interesting to order those genes, order those microRNAs in the same way across the three stages to sort of see if you see a signature for the common microRNAs in them. And sort of a little bit related to that, at the same time you showed those, you were talking about how CCL20 is a chemokine that's really important in respiratory infection. And in these 0 minutes we have left, can you just give a quick, little more detail about that?
So I must say in our studies and a lot of studies, the group that creates that is the hardest one is what we call the moderate disease. So the in-between groups. So you could, as you saw, we could have very clear, and if you look at the paper, you know, there are very clear signatures of mild and severe disease if you compare to the rest of the group. The moderate group seem to be a little bit more tricky to have a list of identifiers. The second, oh yeah, CCL20, there is a lot of literature about CCL20. It pops up in a lot of the studies that were similar, but then, yeah, I think they were even, it has an interesting thing. It's one of these chemokines that only has one receptor, and it's the only chemokine that binds it. That's very unusual for chemokines. There were some therapeutic programs in some of the big pharma that were stopped for some unwanted effects there. So yeah, we could talk about it, but yeah, yeah, look it up. There's a lot of interesting things about CCL20 in respiratory infections.
Lots more questions, but we're at the hour. Thanks so much for talking to us today.
Yeah, thanks for having me. And yeah, if anyone wants to get in touch, absolutely.
Great. All right. Thanks, everybody.
All right. Bye-bye.